Well, we have Dr. Penalva and Dr. Beekman here to talk to us a little bit about a very interesting medulloblastoma project that's been going on, talking about rRNA modification. We wanted to learn a little bit more about this and how it impacts some of the more aggressive forms of medulloblastoma. Dr. Penalva?
Okay, well, thanks for, sharing, giving us time to share our research that is on ribosomal biogenesis, RNA modification in the context of medulloblastoma. We are trying to kind of, learn about this process to see if we can identify novel ways of, target medulloblastoma. So the central idea here is that, you have, cancer cells, medulloblastoma cells, and they need to produce more proteins than normal cells. So they need to increase their translation, and also they need a specialized translation to be able to grow, activate oncogenic pathways, and many other things that, cancer cells need. So how they do this? So essentially you have this structure called the ribosome that is responsible for translation for protein production. And essentially, the idea is that, the cells are going to kind of, re-engineer the ribosome to be more efficient and also to be, specialized to be able to kind of, translate specific RNAs that are involved in oncogenic process. So one thing that is critical for the formation of the ribosomes, for the ribosome biogenesis, is a ribosomal RNA modification. So the ribosome has several proteins and has also the ribosomal RNA, and the ribosomal RNA, it can get modified in very specific positions. And essentially, this is going to kind of like somehow help the ribosome to select proteins, and different combinations can help can occur here, and this is going to affect the composition, the stoichiometry of the ribosome, and how the ribosome is going to function, preferences in terms of mRNA, proteins that get translated. So there are two types of modifications that are called, like, the 2O methylation and pseudodenylation. And very important to kind of, select to drive these modifications are one type of small RNA called SNO RNAs. So the SNO RNAs, they are going to base pair with the ribosomal RNA, and they're going to help select exactly the nucleotide that, gets modified. So there is almost nothing in terms of characterization of the SNO RNAs in brain tumors, specifically in medulloblastoma. So we start, by developing one sequencing platform to specifically characterize these SNO RNAs. And we did a study in medulloblastoma that was funded by the Curious Starts now. So this is kind of, a summary of the most important data that we identified in this analysis. So what I'm showing here are three different studies that we did with medulloblastoma group 3 cells that, there are more aggressive ones, tumors, and other cells of group 4 that also kind of like, very aggressive and express that oncogene MYC. So when we put all this together, these three studies that we did, we see that, there is a pattern here with, I'm kind of highlighting the ones that are common in green and blue. So this helps us to identify SNO RNAs that could be contributing to the process of ribosomal biogenesis, or specifically to these changes in modifications that can drive or can modify the ribosome to be more efficient and to translate the specific mRNAs. So one thing that is interesting is that, how this process is coordinated. And one thing that we identified is that this process is coordinated in medulloblastoma. This process is coordinated by a group of ribosome biogenesis factors that are kind of, tightly connected and regulated by the oncogene MYC. This is kind of, very important because, now kind of, understand the players in ribosomal biogenesis in medulloblastoma, and this also gives us another avenue to kind of, target these specific factors that can kind of modify the ribosomal RNA and produce a different ribosomes. One of these factors is called discarin, DKC1, and this is an enzyme that is responsible, so it's part of a complex together with the SNO RNAs that's important for this for triggering this modification called pseudourylation. So the idea that we have here, so DKC1 is regulated by MYC. You have increased expression, increased levels in medulloblastoma cells. This is going to affect the pattern of pseudourylation in ribosomes, that it's going to kind of affect the formation of the ribosomes and have an impact on translation. So we decided, we start collaborate with Dr. Andrew Beekman, and we developed a specific inhibitors to target discarin-1, DKC1. So Andrew now is going to kind of present like the work he developed to have inhibitors against DKC1.
So yeah, we're a medicinal chemistry group, and we try to think about how to target interesting, important drivers of cancer and control, particularly the protein-protein interactions in cancer. And so after you've identified that DKC1 is a great target, we need to then think about a way to functionally block that. And to think about our targets, we use an approach that tries to take advantage of the genetic mutations that are observed in lots of different diseases and think about how they can be applied to tackling cancer. So for example, you might have a disease which causes some kind of genetic mutations. And the notation on the left here is just highlights those mutations. Those amino acids are changed from one to the other. And you can then map those genetic mutations to the structure of a protein. So this ribbon diagram here is the structures that we use for proteins, and the purple highlights where those amino acids are changed in these diseases. And then if you can, sorry, go to the next one. And then if those mutations appear on the surface of that protein, and they are very likely to impact the protein-protein interactions. And protein-protein interactions are what drive most of biology. Some kind of formation of complexes needs those proteins to come together and build up into those large complexes that might drive, for example, ribosome biogenesis. And so if these mutations cause the activity to decrease because of the inhibition of those protein-protein interactions, perhaps we can use them. And if we think about then in a second disease, for example, cancer, where the protein interaction activity is increased, perhaps by increased expression or enhanced protein affinity, then that protein-protein interaction goes up, and we can use that as a way to guide how to target cancer by targeting the interaction that has been highlighted by that other disease. And so in this case, we do that with a disease called dyskeratosis congenita, which is a premature aging disease where ribosome biogenesis is ineffective. It's not as available, and we've used that idea to try and target cancers. So just on the next slide. So we can see that here again in this complex. This is called the HACA complex, which is the complex that brings together the rRNA and the proteins that do the pseudourylation and change RNA, and is modified in cancer to turn up ribosome biogenesis. And the purple dots are where the mutations exist in that premature aging disease in this protein-protein interaction. And we identify that those mutations are mostly clustering at the boundary of the two DKC1 proteins, shown in green and brown here. And so we could then target that system, and you can see here in the zoom in a small helical section that really drives a lot of the binding between those two proteins. And so we made peptides that were based on that protein section and took in the most common mutations. And this is a picture of that peptide. So the peptide is just a small part of that protein. These letters at the top of the sequence, the amino acid sequence of that peptide. And we synthesized these and then tried to staple them together. So between the two amino acid residues that are on the same side of that helix, we can make a chemical bond, and that makes them stuck into that helix shape and makes them better binders. And so you can see the sequence of the ones we've bound here, and then we can see that those peptides are able to inhibit our protein-protein interaction. They're able to bind to our proteins, and it's done in a dose response. So the more peptide we add, the better the interaction is. And then we were able to observe between the two proteins that we can inhibit that protein-protein interaction. And that last graph F there shows that we're able to make peptides that are more stable once we're stapling them. So the longer they last, the more the downhill that slide is with stapling. So we made those peptides. We identified that they worked on the proteins outside of the cell. And then we sent them to Dr. Penalva to look at the proteins inside those cancer cells and what they can do.
So then like, having like this peptide inhibitor. So the idea that, we want to advance, this peptide inhibitor in therapy. And the first study that we did, we use cell systems, medulloblastoma group 3 cells, and we test them to see kind of, this can inhibit DKC1 and can halt the growth of cancer cells. And this worked relatively well. So one good thing that, in the first assay that I show here, we have 2 medulloblastoma cells, the D458 and MB03. So we see that, this affect the viability of the cells, but didn't affect the viability of astrocytes, normal neuronal cells. So this affect also kind of, the proliferation of the cells, increase apoptosis of the cells, and also protein synthesis, as you can see here, like this is a traditional what we call a puromycin assay. And remember that, you mentioned that, the DKC1 interacts with SNO RNAs. In fact, it is involved in the processing of SNO RNAs and in ribosomal RNA modification. And in the first slide, I showed like several SNO RNAs that are altered in medulloblastoma. So what we saw is that, by adding this inhibitor to the cells, there is a decrease in the levels of this, what do I not say, oncogenic SNO RNAs. And also kind of, affect telomerase formation. This is a second function of the DKC1 protein. So this kind of, gives us, confidence that, this peptide inhibitor that was developed by Dr. Beekman, we can advance this. And now, like, our goal is to kind of like, start medicinal chemistry studies to be able to use this peptide in vivo and start, using them in medulloblastoma models. So in conclusion, so our work, so we have like, 2 main parts, the characterization of the SNO RNAs. And I think, in both cases here, the goal is to kind of, try to develop new alternatives, alternatives for therapy. So the SNO RNAs, now what we're doing now that we characterize the SNO RNAs, we have a list of SNO RNAs that are altered in medulloblastoma. We started to use what is called antisense oligos to get rid of the SNO RNAs and see if this can kind of, halt the growth of medulloblastoma cells. And also, we want to continue with the analysis of the DKC1 inhibitor to kind of, see if we can modify this to allow these inhibitors to penetrate, to cross the brain-blood barrier and target medulloblastoma cells, hoping that one day we can kind of, use this in therapy. We are also kind of like, remember that, I show a list of factors that we identify that are altered in medulloblastoma related to ribosomal biogenesis and driven by the oncogenic MYC. So together with Dr. Beekman, we are now kind of, exploring other alternatives, other factors that we could target, develop inhibitors, and use them in therapy. So yeah, I think this is the end of our talk. Thanks again, like for giving us a chance to kind of present our work.
Fantastic. We appreciate learning a little bit about the trial and some of the work that you're doing with this project. Have a couple of quick questions, if I may. In the grant application, you focus on subgroup 3 and 4 as one of the main concentrations of this concept. Is this something that is effectively just for group 3 and 4, or does it have any applications beyond that?
No, it has applications beyond that. So, for instance, so ribosomal biogenesis at the end of the day is altered in many different tumor types, pediatric and adult. And of course, including like, pediatric brain tumors. So we test actually these inhibitors in other types of cancer, glioblastoma, for instance, and so there is potential use of targeting DKC1 and perhaps other proteins in ribosomal biogenesis in other types of pediatric brain tumors. The same is true for the SNO RNAs. So we focus our analysis on group 3 and group 4, but we did an analysis in other tumors. Glioblastoma is one of them, and interestingly, we identify several parallels. So there are several SNO RNAs that are not only altered in group 3, group 4 medulloblastoma, but they are altered in glioblastoma and other tumor types. So this means that the strategy that we are trying to develop to target now these SNO RNAs that are altering medulloblastoma, but other tumor types, we can use the same antisense oligos to perhaps target other pediatric brain tumors.
Okay, fantastic. You also talked about the applications to try to move this to a therapeutic effort. Obviously, there's some work that you're still going through here with it. What's your expectation on when do you think that might be available for a therapeutic option?
Yeah, I think, Andrew can comment on that, because now, this is happens to any molecule that you have, that you have the in vitro and the cell assays moving them to to in vivo and be able to use in therapy. You have to do medicinal chemistry that, Andrew can elaborate.
Yeah, so we've this is really the first proof of concept that targeting this system has a really positive effect on medulloblastoma. And so those compounds now, they need to undergo some changes to make them more clinically relevant. One of the big challenges of this is that they'll need to be able to cross the blood-brain barrier. And so we're working on developing modified versions of these compounds that are kind of like Trojan horses that we can convince the cancer cells to take up and then modify to then release that drug, just in place, just where the cancer cell is. That takes a campaign of chemistry to go and test those things and make sure that they're still as effective. That's work that's ongoing in our lab, and hopefully we'll have some exciting more results out of that in the coming months to be able to have a clearer timeline, I guess, on putting that into in vivo into animal-based models maybe.
Yeah, and
Right.
also want to say that besides the peptide inhibitors and develop small molecules, and that also we started to test that will be also they target DKC1, and it will be another alternative, perhaps easier to modify and you feel like some of them be able to cross the brain-blood barrier without having to we have to add any additional modifications.
Well, perfect. Dr. Penalva, Dr. Beekman, thank you for talking a little bit about this. That's the whole purpose of this, is to get the word out about the great research that you are doing. And we're excited to see some of those next steps.
Well, thanks so much.
Thank you.